Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore one of the most ingenious experiments in the history of biology, the CLB method in Drosophila. Imagine trying to find a tiny, invisible bomb hidden inside a massive library of millions of books. You cannot actually see the bomb, but you know it is there if a room suddenly collapses. In the early days of genetics, scientists faced a similar problem. How do you detect a genetic mutation that is completely invisible and kills the organism before it is even born? Let us dive into how a brilliant scientist solved this exact puzzle. To understand why this method was created, we first need to understand the problem early geneticists faced. Before modern DNA sequencing, scientists strongly suspected that outside forces like radiation could damage living things. However, they had absolutely no reliable way to measure exactly how much genetic damage was being done. If a mutation is so severe that it kills a fruit fly before it even hatches from its egg, you cannot simply count dead flies. They just do not appear. Therefore, researchers desperately needed a special detector, a genetic sorting machine that could clearly reveal when a hidden, deadly mutation was created by radiation. This brings us to the brilliant solution developed in the 1920s by scientist Herman J. Muller. He designed a clever genetic trap known as the CLB method. This classical genetic technique was specifically created to detect induced sex-linked recessive lethal mutations in the common fruit fly, Drosophila melanogaster. Instead of trying to look at the DNA directly, Muller engineered a very special X chromosome that acts as a built-in alarm system. By tracking this specific chromosome through 2 generations of carefully controlled breeding, scientists could definitively prove whether a mutagen, like X-rays, caused a fatal error in the fly's normal DNA. Now that we know what the method does, let us break down the first 2 crucial elements of this engineered chromosome. The name CLB refers to specific genetic modifications. The C stands for a crossover suppressor. This is a large chromosomal inversion that prevents genetic recombination from mixing up the genes during cell division, ensuring our detector stays perfectly intact. The L stands for a recessive lethal gene. Because male flies only have 1 X chromosome and cannot mask its effects, any male inheriting this specific gene will automatically die. Together, these 2 elements create a stable, deadly trap that is ready to catch new mutations. Let us take a closer look at one specific part of that chromosome, the bar I marker. You might wonder how scientists know which flies actually carry the special CLB chromosome if the lethal gene itself is invisible. This is where the B comes in. The bar I is a dominant physical mutation that physically alters the fly's appearance, giving them narrow, slit-like eyes instead of normal round ones. It acts as a visible tag for the invisible genetic trap. Because it is dominant, researchers can simply look through a microscope and easily pick out the exact female flies that need to be selected for the next round of breeding. Without this visual marker, the entire experiment would be impossible to track. With our special chromosome understood, let us look at how the actual experiment unfolds across generations. After exposing normal male flies to a mutagen, they are mated with CLB females. In the first generation, researchers specifically select the daughters that have those narrow bar eyes, because they carry both the CLB chromosome and the irradiated chromosome from their father. When these females breed to create the second generation, the magic happens. Any male inheriting the CLB chromosome automatically dies because of the built-in lethal gene. Therefore, if the other chromosome, the irradiated one, also acquired a lethal mutation from the experiment, all the remaining males will die too. A vial with 0 males means a mutation was successfully caught. To make this completely clear, let us walk through the visual diagram of this genetic cross. At the top, in the P1 generation, we see our irradiated normal male being crossed with the CLB female. Moving down to the F1 generation, we select the bar-eyed female, who now carries 1 irradiated X chromosome and 1 CLB chromosome. We cross her with a normal male. Now look closely at the bottom row, the F2 generation. We have 4 possible offspring. The CLB male automatically dies, crossed out in red. The critical part is the irradiated male on the far right. If the original radiation caused a lethal mutation, this male also dies. The result is a culture vial containing only female flies, which is our definitive proof of genetic damage. Seeing this elegant genetic cross in action brings us to the massive historical significance of this method. Before this experiment, scientists could only guess about the dangers of radiation. The CLB method provided the very first quantitative, mathematical way to measure mutation rates. It proved definitively, without a shadow of a doubt, that X-rays cause severe genetic mutations. This discovery was so monumental to human health that it earned Herman J. Muller the Nobel Prize in Physiology or Medicine. Today, whenever you wear a lead apron at the dentist or follow chemical safety protocols in a lab, you are being protected by the foundational knowledge of DNA damage that was first proven using these tiny fruit flies. Now that we understand the biological mechanism and its importance, let us look at the mathematics behind it. This formula allows us to calculate the exact rate of genetic damage. The symbol mu stands for the induced mutation rate, which we express as a percentage. The variable NL represents the number of lethal cultures. These are those specific second-generation vials we just talked about that completely lack any male offspring. The variable NT represents the total number of second-generation cultures that were successfully bred and examined. By dividing the lethal cultures by the total cultures, we find the fraction of mutated chromosomes. We then multiply by 100 to get a percentage. If the mutagen is very strong, NL increases, and the overall mutation rate, mu, will proportionally increase. Before we move on, let us see this formula in action with a concrete example. Imagine a researcher exposes male fruit flies to a controlled dose of X-rays. Out of exactly 500 individual second-generation cultures evaluated, the researcher observes that 15 of these cultures have absolutely no male offspring. We're asked to calculate the induced mutation rate. First, we identify our given values. The total number of cultures is 500, and the number of lethal cultures is 15. We substitute these directly into our equation, dividing 15 by 500. This gives us a decimal of 0.03. Multiplying by 100 converts this to a standard percentage. The final answer is 3%. Physically, this means 3% of the sperm cells from the original irradiated flies sustained a fatal genetic mutation. Let us tackle another example, this time testing a newly synthesized chemical compound. A laboratory sets up a massive screening experiment involving 1,200 separate cultures. After breeding, they find that 1,128 cultures contain a healthy mix of males and females. The rest contain only females. We need to find the mutation rate. Notice that we are not directly given the number of lethal cultures. We must first subtract the healthy cultures from the total cultures. 1,200 minus 1,128 gives us exactly 72 lethal cultures. Now we plug 72 into our formula as the numerator, divide by the massive total of 1,200, and multiply by 100. The result is 6%. This indicates the chemical is a highly dangerous mutagen, causing double the damage of our previous X-ray example. We have covered a lot of ground today, so let us summarize the most important ideas. First, the CLB method is a foundational screening tool designed to detect invisible, deadly mutations. Second, it relies on a brilliantly engineered X chromosome that uses crossover suppression, a built-in lethal gene, and a visual bar eye marker to track inheritance. Third, the entire experiment hinges on observing the second generation. If a culture vial completely lacks male offspring, it definitively proves a lethal mutation was induced. Finally, this elegant technique provided the very first mathematical proof that radiation causes genetic damage, forever changing our understanding of biology. You now understand how classical geneticists solved incredibly complex puzzles long before modern DNA sequencing was ever invented. Great job following along.